students, so we're going ahead with question number 8. Question number 8 is a difference, and we are going to do difference in two ways. One, the classical way of writing the difference, and the other one, we're going to categorize the difference. Now, we'll start with A. A is repetitive DNA and satellite DNA. Now, for repetitive DNA, it is a sequence of nitrogenous base present in more than one copy of genome. Just remember, students, just like its name, repetitive, that means repetition. So, there is more than one copy of the bases in the genome. Now, satellite DNA is a part of DNA having highly repeated short sequences of nitrogen bases. So, satellite DNA is a part of DNA. Okay. So, what the major difference is? This is a sequence that is present in more than one copy in the entire genome, whereas satellite DNA is a part of DNA. Now we'll go for length of it. So, length of repetitive DNA varies from few nitrogen bases to several hundreds of it. Okay, so it is a long chain. Now, satellite DNA has length of, sequence of satellite DNA is short, and it is mainly 1 to 60 base pair long. Now, repeated DNA sequences may or may not be present in tandem. Tandem means one after the other, whereas in satellite DNA, repeated sequence of nitrogen bases occur in tandem repeat. Now, it does not separate during density gradient ultracentrifugation. Okay, so satellite DNA gets separate during ultracentrifugation. Okay, so in ultracentrifugation is, you take, this is an entire DNA or genetic material. Now, ultracentrifugation is, it will rotate so fast that whatever is present in the genome will start getting varied. So, the highest density gradient molecule will settle down and the lowest one will settle up at the surface. Now, repetitive DNA will not get settled or separated during density gradient, whereas satellite DNA would be separated. Now, variability may or may not be present, whereas in satellite DNA, variability occurs. It is present, but when is it present? It is present when there is misalignment of chromosomal pairing. Okay. Now, let's categorize this. We've got categories, repetitive and satellite DNA. So, the sequence. Repetitive DNA has more than one copy found in the genome, whereas satellite DNA is highly repeated. Length, a bit long, and this is short sequences. How long? Students, 1 to 60 base pair. Now, tandem repeats may or may not be present, whereas they are always present. Separates while ultracentrifugation? No, yes. Variability may or may not be present, whereas it is present. Okay, students, so this is how you're supposed to write a difference. So, we are done with difference number 1. Now, we are going to difference number 2. Difference 2 is between mRNA and tRNA. Now, mRNA stands for messenger RNA. tRNA stands for transfer RNA. So, mRNA is large in size, whereas tRNA is clover leaf-like small sized RNA. Okay, so tRNA will look something like this. And then there is also this. So this is how a tRNA looks. So it is what? Clover leaf like small sized RNA. Now mRNA carries coded information, whereas tRNA carries information for association with an amino acid anticodon for its incorporation in a polypeptide. Now tRNA will carry what? tRNA will carry an anticodon so that it can transfer information. Now mRNA undergoes additional processing like capping and tailing, whereas tRNA possesses 3 to 4 loops and limb distinct 5' to 3' or 5' to 3' end. Nitrogen bases are unmodified. Nitrogen bases are what, students? Nitrogen bases are not modified, whereas in tRNA nitrogen bases are modified. Okay, now let's categorize this. Now categorizing mRNA and tRNA. Size large, clover shaped. Information coded has an anticodon. Now additional processes capping and tailing. tRNA possess what? 3 to 4 loops and a distinct tail and has 5' to 3' end. Now nitrogen bases unmodified and they are modified in tRNA. So we are done with difference number 2. Going ahead with difference number 4. Difference number 4 is template and coding strand. Template means a reading strand. Okay, so let's see. Template. It is a strand of DNA which takes part in transcription. Remember, it takes part in transcription. Coding strand will not take part in transcription. Polarity is from 3' to 5'. Template is 3' to 5'. In coding strand, polarity is opposite to this one. It is from 5' to 3'. Now, nucleotide sequence is complementary to one present in the mRNA, whereas in coding strand, nucleotide in mRNA except for presence of T, we have got U. Okay, students? So this is a basic difference. Now, let's categorize this. So we'll write categories, template strand and coding strand. Takes part in transcription. Yes. Does it take part in transcription? No. Polarity 3' to 5', whereas coding strand would be 5' to 3'. Now, nucleotide sequence completely complementary, whereas for coding strand only T is absent in the place of U. So for mRNA we'll have U instead of T. So T is replaced to U. Remember this point. This point is important. Okay, students. So we are done with our difference number 3 as well.